Good afternoon. Welcome everyone for today's endocrinology grand rounds. We are pleased to have Dr. Shirish Barve present to us today. Dr. Barve is professor of medicine and distinguished university scholar at the University of Louisville Department of Medicine and the Division of Gastroenterology and Hepatology. He's also the chief research scientist at Norton Neuroscience Institute and the director of the Norton um, Research Institute. He obtained his Ph.D. from University of Bombay and sorry, he obtained his master's degree from University of Bombay and came to, obtained his PhD from the University of Kentucky in Lexington, where he had worked with Dr. McLean, and in 1999 moved to University of Louisville, where he has been since then. His main interest has been in the study of liver disease and inflammation and under various different circumstances, and particularly with relation to alcoholic liver disease, fatty liver disease, HIV-related liver disease. And in the process of studying these, he developed a great interest in the study of the gut microbiome and how it interacts with the liver disease. So initially, a lot of focus on the gut-liver axis. And of course, this further led to his interest in the gut-brain axis. Actually, we may call it the gut-liver-brain axis. And he has been an international leader in the study of the gut microbiome and how it interacts with the gut-brain axis, and has been funded for his research over the last 25 years or more years continuously by the NIH with several R01, UO1, and P60, and multiple grants. In addition to that, also from the Department of Defense. And so he has been well funded over all these years. He has mentored many PhD students and also many of the residents and fellows who have worked with him in the lab during their training here. He has over 150 publications. His current interests have been related to the gut-brain axis. We have had some of our fellows work with him in the past, and there is an overlap between the gut-brain axis and the endocrine system. And of course, if you have listened to some of the radio, internet, and podcasts, many people will call the Alzheimer's as type 3 diabetes, So, of course, it is related to insulin resistance. So I'll have him present to us some of his latest work that he has done in the field of the microbiome and the gut-brain axis.
I was going to say there's water there for you.
Thank you very much, Dr. Mokshabindam, for the kind introduction, and thanks for the invitation to share our recent work. As the title says, I'm going to talk about the relevance of the microbiome-gut-brain axis in the onset and progression of Alzheimer's disease. And our byline is fix your gut and fix your brain. But I'm going to do this in the context of the metabolic issues that surround Alzheimer's, including type 2 diabetes. Very brief background. This is the picture of a descending colon. Starting from the stomach, there are very few bacteria because of very low pH in that environment. And as you go down in the system, the number of bacteria start growing. Till you reach the colon, you're looking at roughly a few trillion bacteria. So it's a large compendium of microflora that sits in your GI tract, and it continuously communicates with your system, directing both health and or disease. So this is another trivia is human genes. We run on roughly 23,000 genes that make us who we are. But in your gut microbiome, there are roughly million plus genes. So it's a huge genetic information that again sits there. So this is a liquid organ that is there that continuously talks with your system and clearly plays a significant role. Before we go into its dark side or the bad side or the disease producing side, your microbiome absolutely plays several beneficial functions in relation to your system and maintaining your health. So first is clearly the metabolic functions where there is fermentation of non-digestible substrates. That's what happens when you have your greens and non-digestible fiber that goes in your diet. There's bile acid biotransformation, very important. Vitamin production, energy extraction. Protective function. This is clearly what protects us from the bacterial and viral infections, including some of the evolution of cancers. A friendly or an appropriate gut flora is highly protective. There is colonization resistance. And what that means is we encounter pathogens on a daily basis, but your commensal flora, if it's appropriately managed and maintained, it will not allow these pathogens to establish and then gain a foothold, protecting you from infections. Antimicrobial secretions, bacteria, again, your commensal flora will make bacterial secretions that are unfriendly to the incoming pathogens, and that's how you get protected. And most importantly, the microbes continuously shape and reshape your innate as well as your adaptive immune response. And there's one more slide, as we go down further, you'll see this more clearly. But this is where you can either start some chronic inflammatory issues or you can get highly protective issues that emanate from the gut. There are structural and histological function. For example, the right kind of host microbe interaction controls the epithelial cell growth and differentiation of the intestinal epithelial cells. Intestinal villi and crypt development is influenced by the right type of microbiota, and most importantly, the tight junctional permeability. So you are as strong as your gut, as they say. You have heard the terminology leaky gut. So tight junction proteins like zonulins, claudins, or occludins are again significantly shaped and reshaped, and their functional aspects are also controlled by the microbe and the host interaction. So these are some of the highly beneficial functions that your microflora offers you. In the context of current topic, I'm just going to point out the burden of Alzheimer's disease. It's a pretty devastating statistic, as you can see. Right now, there is 1 in 3 adults have Alzheimer's. It kills more than breast cancer, prostate cancer combined. There is a significant rise in the incidence compared to some cancers. But most importantly, the cost to the U.S. healthcare is staggering. Right now, it's at about 384 billion, and it's about to touch $1 trillion by 2050. And on the right hand, so these are the national statistics. On the right hand side, you have the Kentucky statistics. And just a few months ago, when I had a slide, it said that the Medicaid cost in Kentucky was at about 870 million. And this is the latest. Now it has touched $1 billion already, Medicaid cost in Kentucky. So let's start with some background about this disease. These are this is a classical kind of a cartoon representation depicting autopsy studies which show that most people develop some plaques and tangles as they age. We all have that in us. However, individuals with Alzheimer's tend to develop far more plaques and tangle burden, and in a predictable manner, beginning in the areas important for memory, like the hippocampus out here, and then starts spreading as the stages of mild cognitive impairment, advanced dementia, and then Alzheimer's develops, as is shown here. So it grows in stages. I will make a comment later on that the Alzheimer's disease biomarkers, which we are now able to track, appear in us 8 to 10 years before the diagnosis is made. So this is something we will discuss towards the end of my talk. This is the neural basis of Alzheimer's disease. So though I would mostly function to begin with, we talked about the plaques. So these are made from these are A-beta plaques from APP protein, which is processed inappropriately by a protease called presenilin. And then these sticky clumps of A beta peptides form extracellular clumps, and which are the plaques. And this is the plaque burden that starts or keeps increasing. But even more relevant, as we now know, are the neurofibrillary tangles. And this is a normal neuron, and these are the tangles depicted here. That's a cartoon, which are formed by the tau protein. So the normal function of the tau protein in the brain is to stabilize the microtubules, which are essential for maintaining the structure of the neurons and for transporting nutrients and waste. This is an extremely important component. But when tau starts getting hyperphosphorylated because of inflammation, oxidative stress, genetics, epigenetics, all those things, they leave the microtubule and they start forming neurofibrillary tangles. These neurofibrillary tangles are extremely neurotoxic, and they start hurting the neurons. There is a large amount of neuronal apoptosis, loss of neurons, and as you can see, there is a significant loss in the volume of the brain because of all these events. So you have the healthy brain in this part, and then you have the severe Alzheimer's brain, and obviously there are stages in between. So this is the hallmark pathology of this disease. The typical progression of cognitive decline in Alzheimer's disease is shown over in this graph here. This is the normal aging. We all have issues. When it comes to memory, by the way, there are two components. One is memory formation, and the other is memory retrieval. So it's important that you can have deficits. Usually, memory retrieval is affected as a part of normal aging. We don't remember names, we don't remember, but it's not going to impact our day-to-day functioning. Whereas in the context of Alzheimer's, the preclinical Alzheimer's brain changes start decades, as I said before, symptoms show. And now we have biomarkers, even plasma-based biomarkers that we can track, progressing into mild cognitive impairment, where brain compensates for the changes, and then eventually slips into dementia, where there is cognitive decline and loss of independent function. And then there's a diagnosis of AD dementia or Alzheimer's disease-related dementia. By the way, there are many other forms of dementia. I'm going to focus on Alzheimer's disease-related dementia, which is about 60 to 70% of the overall dementias is predominantly Alzheimer's. This is the PET scan to show how a normal brain, mild cognitive impairment, and Alzheimer's disease. There is significant loss of brain volume. And all this is accompanied by loss of cognition. So there was recently 21-22, the Lancet Commission basically conducted a research which revealed that 45% of the cases of dementia can potentially be delayed or reduced. So these are all modifiable risk factors. And pertinent to our audience today, I've highlighted the issues which are metabolic issues, which are part of the modifiable risk factors connected with Alzheimer's. So you have obesity, hypertension, diabetes, and high LDL cholesterol. All these, there is hard evidence to show that if these are addressed, there can be a significant aspect that can lead to prevention, or at least a delay in the onset of dementia. So this is something very significant. So as Dr. Mokshinda mentioned, I joined Norton as a joint appointment to basically examine, because I'm going to the genesis of that, to examine the issues connected with Alzheimer's and the potential role of the gut microbiome. So this is a clinical translational program that we have. The basic intent is to be able to inform clinical practice. And as you can see, the cartoon says we have the questions we are addressing, identifying at the bedside. We are bringing them to the bench side, hopefully developing answers, solutions, taking them back, and ultimately taking it back to the community for improving healthcare. So that's the overall program in a nutshell. So this is the 10,000 feet overview of the overarching hypothesis that we are working on. So that the pathogenic changes in the microbiome gut-brain axis plays a major role in the development of Alzheimer's disease. So if you look at microbial dysbiosis, this is a term that is always used. And there is microbial dysbiosis in myriad of disease states, from type 2 diabetes to colon cancer to depression to anxiety, all kind arthritis, you name it. And what that means is you're changing your commensal flora, and it's changing the composition of this commensal flora. A significant aspect when you start changing the composition is you start losing diversity. So the microbial diversity changes. We have 500 plus species, and you start losing this diversity. You're really good health is connected with a very diversified microflora that protects you. When you start losing diversity, what can happen is there is a gain in potential gain of pathobionts or disease-causing bacteria. And the flip side of that is that you start losing beneficial bacteria. And that's you the end game that what we have seen with Alzheimer's and also with type 2 diabetes is that you're losing beneficial bacteria over time. That is playing a causal role, and you'll see evidence that I'll present. But bottom line is, right from the mode of delivery, normal delivery versus C-section, level of stress in an individual, sleep, extremely important or lack thereof, fragmented sleep is truly an important component that drives the issues of dementia. And that's because the way brain unloads its toxic components, it happens mostly due in your deep sleep. That's why if you have fragmented sleep, your ability of the brain to unload or detoxify is compromised. Diet, obviously connected to all the metabolic issues, high salt, high fat, how much alcohol you drink, and so on. Environment, there are very strong connections to particulate matter pollution, other types of pollution. Where does an individual live in the countryside, in the inner city, and so on. Antibiotics or polypharmacy, particularly with elderly patients, they're on multiple medications. Anything that you ingest, your microbiome does pay a price for that. And then obviously normal aging. If you had taken our microbiome 10 years ago and looked at it, it's not the same today after 10 years. So overall, what we have is that the microbiome that we have integrates life experiences. So what all goes through us, your microbiome starts changing in response to that, and obviously it is interacting with our system. So before I show you what's going on with the microbiome in the context of Alzheimer's, I thought I would quickly mention this component, that the type 2 diabetes associated dysbiosis is one of the main features is loss of butyrate producing bacteria. So there are several independent metagenomic studies that have detected a decrease of butyrate producers in stools from individuals with pre-diabetes and type 2 diabetes compared to individuals with normal glucose tolerance. So what you're seeing here is a streeter torter where some of these species have been identified. These are large genera of butyrate producing bacteria, which have been clearly documented to be down in a statistically significant fashion. There is some controversy about the butyrate levels because butyrate is extremely difficult to track. It's very rapidly consumed when it is formed. And there have been butyrate replenishing interventional trials reported for type 2 diabetes with benefit. So I won't cover all that. I just wanted to have you this particular component in mind. And now we'll get into the preclinical part, and then I'll talk about the clinical part of the Alzheimer's disease and microbiome. So we wanted to investigate, first of all, the preclinical level, the pathogenic role of the gut microbial dysbiosis. And to that end, we use this triple transgenic mice, which has 3 genes. It has the APP human gene, which APP gives rise to all the A beta plaques. The presenilin gene, which is actually the mouse gene. APP is the human gene. The presenilin is the protease that cleaves APP inappropriately and gives rise to those sticky plaques. And then tau, which is it's a mutation associated with human tau pathology, but basically is expressing the human tau in these mice brains. The most important advantage of this animal model, it's a long model. That's why people tend to use shorter models, but those are more genetic. And in real life, the genetic aspects in patients is about 5% population, whereas 90 to 95% are all sporadic cases of Alzheimer's. And so this is a great animal model because it develops age-dependent cognitive decline, accumulation of plaques and neurofibrillary tangles are formed, and there is age-dependent inflammation. So this animal model, and this is a time trajectory that is documented up here, allowed us to investigate the changes both in the microbiome as well as the brain pathology, as well as the behavioral or the memory pathology that develops. So the main reason to use this animal model was to investigate the causal role. There have been many cross-sectional studies, and yes, there is this biases in a person with Alzheimer's, but you don't know if it is playing a causal role or it's the effect of the disease process. So we wanted to investigate the causal role of the gut dysbiosis in the development and progression of AD, and then to determine the impact of evidence-based management of gut microbial dysbiosis in the prevention and treatment of Alzheimer's disease. So we wanted to identify the features that develop over time and whether those features, if they're addressed, what happens. So this is the first data slide. Loss of butyrate producing bacteria was a significant feature in these animals. So what you're seeing here is a loss of butyrate. So our lab was the first group to report and develop a very comprehensive dictionary of butyrate producing bacteria. There are about 243 genera now. We have published this. So we could express the gut microbiome in the context of total butyrate producing families in a quantifiable fashion. And as you can see, this is a 2-month animal and 12-month, and by 18 months, what we saw, I'm not going to go through other microbial changes, but the most prominent and statistically significant change was a loss of this collective of the gut butyrate producing bacteria in these animals. And it's shown here in a qualitative form. These are the families from Ruminococcaceae, Oscillospiraceae, and so on that, as you can clearly see, are changing over time. And connected with the structural changes in the microbiome, we also saw a very significant change in the butyrate levels. That means the functional part of this aspect of dysbiosis. And here you're seeing a loss levels of cecal butyrate in these animals between 2 months and 18 months, and there's a significant loss of butyrate as well. So coming back to this cartoon that explains that if you look at this part, which is a normal colon, this is a normal looking intestinal epithelial cell line. This is the mucus layer. You have healthy bacteria. This is your fiber, which is non-digestible, acted upon by Faecalibacterium prausnitzii in this cartoon. And then you get butyrate. And what you have surrounding your alimentary canal is a loose connective tissue called lamina propria. And it is here where your immune cells keep coming. That's connected to your circadian rhythms as well. So this is where you have all the monocytes and macrophages and adaptive response like specialized T cells and B cells, and they're all there, and they are literally there to slurp what's going on in the gut. And I guess that's why the adage, you are what you eat. But this is where your immune response is getting trained in a good way or in a bad way. And then obviously they can leave this. First organ that sees this is the liver through the enteroportal circulation, and then all these things start changing. Here is on this side a pathologic situation. It could be diabetes. It could be in Alzheimer's. You're finding similar things. Is you are losing this mucus layer. There is paracellular permeability, and your microbial antigens or intact microbes gaining access. And these are then serve as PAMs, or pathogen-associated molecular patterns, that significantly drive your immune response crazy. And you start this smoldering inflammatory situation that goes on. Pardon me? Yes, yes. So, but the microbial antigens, once your immune response sees that, that's how we have evolved, is to sense these PAMs. And they start making inflammatory molecules. Your adaptive immune response changes. Many things happen. So because we saw, sorry, how do I go back? Yeah, because of the butyrate storyline, again, not going into the whole genesis of the rationale, we decided to examine the effects of tributrin, a butyrate prodrug, on the pathogenic changes in the microbiome gut-brain axis. So we just decided to check this out. Tributylin, by the way, is present in dairy. The butteriness of butter is because of tributylin. It's a glycerol backbone, and 3 molecules of butyrate are condensed on that. So when tributylin is there in a dietary form or taken orally, your pancreatic lipases, as well as the right kind of friendly bacteria, can hydrolyze that and release butyrate in the system. Giving sodium butyrate was not an option. That caused hyponatremia and has other issues. And this was the best possible way to give, replenish, and ask the question, what would happen? So what we did was we started painstakingly giving animals at about 6 months tributylin, oral gavage, for 18 months. So this was a long experiment because we decided, because starting, the data showed that starting 6 months, the bugs are starting to change with age in these animals. This is the first data set. So this is your 18-month animal where it has, these are the butyrate producing families again being tracked. And these are the same 18-month-old animals that were given tributylin. You can see a remarkable difference that tributylin did not allow the butyrate communities to go down, which was actually a serendipitous finding because we didn't expect butyrate producing bacteria to be protected and preserved. We thought that we are just going to supply the butyrate, which is being lost. And that's being, it's a quantifiable measure here, very significant data points in these animal experiments. Let's see the consequences at the level of the brain. So most importantly, oxidative stress is a very significant component of Alzheimer's disease. And 4-hydroxynonenal, which is a lipid peroxidation byproduct, plays a predominant role because your brain is a very lipid-rich organ, and oxidative stress leads to lipid peroxidation. And we saw that the 4-HNE was markedly attenuated here at the same age, but that animals that got tributylin starting at the age of 6 months. Another very important oxidant shown in spinal cord injury, in any other neurological issues, is acrolein. It's a very reactive aldehyde, and again that was also very significantly dropped by animals receiving tributylin. And by the way, butyrate is known to have antioxidant effects and so on. This was the most important result also, where we looked at basically hyperphosphorylation of tau. As I mentioned, that this is the key pathologic event, that neurofibrillary tangles, because they have shown centenarians have plaques, cognitively completely competent, but it's a tau pathology or hyperphosphorylation of tau seems to be of extreme value, and now is considered the pathologic hallmark of Alzheimer's. And this event, which is shown, this is a confocal microscopy, you can see a bunch of action happening here, and this picture is very calm. And as you can see, it was almost completely abrogated in these animals that received tributylin. So the whole cascade of oxidative stress, inflammation, all that was held back by those animals that got tributylin starting at 6 months. So before I go to the next data slide, I just want to mention that butyrate is an extremely important natural HDAC inhibitor, histone deacetylase inhibitor. So it has a huge influence on the epigenetic regulation of gene expression. And this becomes a very important mechanistic focus point in our work. If you can see here, I think yes. So we are now staining, this is the hippocampal region that you're seeing there. This is where your memory neurons, for lack of a better word. And if you can see in a 2-month animal, we are looking at histone 3, which is acetylated at lysine 9 and lysine 14. And what that says is that this is a transcriptionally permissive histone modification. That means genes or promoter regions that have that kind of modification, they're open for business. And they transcribe well and so on. So this is a significant epigenetic control pressure point. And as you can see, by 12 months, it's like the lights are being turned off, they're being dimmed. We are losing. So if you look at the level of histone acetylation in the 2 months, it's down. That's the 18-month animal. The lights have been almost completely turned off if you look at the 2 months. But look at the same age animals that received tributylin. These lights stayed on, and it was amazing. And we are doing all kinds of RNA-seq and all that to figure out, because it allows us also to look at the compendium of those genes that contribute to your memory and so on. No, these are histone acetylation patterns, and each green dot is a neuron. So that's the hippocampus or hippocampi, and this is where your memory neurons are. If the very first cartoon, if you remember, this is where the disease starts. You start having issues of plaques and fibrillary tangles in the hippocampal region, which then spreads and takes over. So these neurons play a significant role. As you can see down here, we are seeing these changes. By 18 months, it's down. It's a 2-star significance. But the animals that received tributylin, that did not happen. These histones were acetylated. So butyrate acts as HDAC inhibitor. What that also, the flip side of this is that Alzheimer's disease is associated with dysregulation of histone deacetylases in the brain, particularly in the neurons. And we are working on that right now. What happened was we then took and did a Spearman correlation. We took all the animals with Alzheimer's, without Alzheimer's, and did this and tracked the total butyrate producing families that is seen here, and we tracked the histone acetylation status. So irrespective of the disease state, if there was a good compendium of butyrate producing bacteria, the histone acetylation was up, irrespective of. And I think that's and you'll see later on what happens to memory in a minute. So that is quantified here. This is the acetylation status that is plummeted, prevented by tributylin, and that's the Spearman correlation that I was talking about. The next step was to find out what happens to, so there are significant changes in the neuropathology that I showed you, connected with what's happening with the gut. So we want to look at the memory function. What time is it? Okay. Okay. So this is an animal model where short-term memory clearly is a significant component that is affected in Alzheimer's patients. So when we see patients in the memory clinic, they remember what is 20 years ago, but they won't know half an hour whether they took their medication or not. And that becomes an issue. So short-term memory. And when I got into this, I did not realize that you can track memory so accurately in animals. So this is in collaboration that we did this. So this is called the novel object recognition test. The animals are kept in the cage that are habituated to the environment, and then there are two objects to which they are introduced to. The animals will go to each object, and everything is recorded by heat sensing camera and quantified. And once they get trained, one of the objects is removed and is replaced by a novel object, which is called a different object. And instinctively, the animals gravitate towards a new object and they spend more time there, as long as they are normal. Once they start developing memory deficits, they don't care whether there's a new object because they can't discriminate. And that is what is documented in the next slide. So a little bit of a busy slide. And these are all the heat tracings. But this is the NORT is a combination, what we call as discrimination index. That means the animal discriminates between the objects, the recognition index, and so on. And this is the NORT quantification that is shown here, which is 10 months, and that's the 17-month animal. And look at what happened to the same age of animals, but that had received tributylin. It could completely preserve the short-term memory. And it was quite a remarkable finding. Was happy to say that this was, by the way, published in Gut Micro, was a pretty high impact, and they took it. The editorial comments are that, well, when are you going to take this into clinic? So, so this was very exciting. What you see in C and D is, in short, is spatial memory. So they can come into the neighborhood, but they can't find their street because we take it for granted. We know where the elevators are, and it's a spatial memory that we have. And they do this in a Y maze. So they'll open the Y and they go in each arm. They'll close one arm. They only go in one side. And then after a while, they open the second arm. The animals which are normal go to the new arm because that was closed. So they now explore that. But when they start getting issues with memory, that drops and they don't. So that's shown here, for example. And then that also was absolutely preserved in these animals. I don't know if I'm showing yes, I am showing that. Another serendipitous finding in this, which I published, is we we saw that frailty was preserved, was prevented. So neuromuscular connections have a big role in playing that control or regulate frailty. And that's a big thing. Now, in fact, what they're showing that the cognitive decline is one-to-one correlation with frailty. And so this was something very significant. This was, as I said, serendipitous. The grip test is, the animal is put on a mesh and you pull it by the tail, it grabs it. The force in dynes is recorded. And here you can see the difference at 17 months where they have Alzheimer's versus the animals that were given tributrin. And this is the rotor rod. So rotor rod is where they not only have to balance by gripping and also balance, so otherwise they fall off. And you can see the Alzheimer's animals are dropping off very quickly. But look at the difference between, again, the 17 months and animals that received tributrin. So it was clear that even the neuromuscular issues, and we are looking into that. One of the collaborators wants to look at sarcopenia because putrid is known to counter sarcopenia, so the muscle strength. So this was very satisfying. And again, we went to the Spearman correlation. We took all the animals, looked at all the total butyrate producing families and tracked the memory. So clearly, irrespective of the disease, if you just focus on the memory component of it, the higher the level of butyrate producing genera were present, higher was the memory score. So this is something very important. So this is published. We presented this in the international conference. It was received really well. But the preclinical finding basically identified that loss of butyrate producing gut bacteria plays a causal role, and we could use this verbiage that the reviewers allowed us in the onset and progression of Alzheimer's disease, because this loss that I showed you earlier of butyrate producing bacteria preceded neuropathology and behavioral pathology. But most importantly, treating the loss of butyrate producing gut bacteria prevented the age-dependent loss of memory and development of brain damage associated with Alzheimer's disease. So this is the preclinical work. We're doing other studies, like we are looking at, in fact, it's part of the P50. I think it's going to get funded if NIH stays alive. Where we are looking at how alcohol exacerbates, because that's part of my alcohol center thing, which I do with McLean. So that's that. So right now we are pursuing longitudinal studies at Norton and randomized control clinical trials in a well-defined patient cohort that is targeting the gut microbiome with the goal of developing novel preventative and therapeutic strategies. We are also developing systems biology approaches to generate clinically relevant data from clinical cohorts with Parkinson's, Alzheimer's, bunch of other neurodegenerative diseases to guide and establish individual treatment selection and improving patient care. Just as an aside, we are also examining patients that are receiving lecanemab, which is the FDA-approved therapy now. It doesn't cure Alzheimer's, it delays the onset of Alzheimer's. But our thought is that your microbiome or the state of microbiome also imposes on treatment outcomes, which is of high value. So right now you get about a 26 to 30% delay in the onset. If you can push that number to 65, 70, so the thought is don't change the standard of care, but aggressively manage the gut, then that might have very beneficial outcomes. These are the clinical trials. Let me go to the human study. What time is it? Oh, okay. I'll go through this pretty quickly. So these are actual data, not published. It's just happening from our memory clinic. We are looking at inflammation, particularly MCP1 IP10, which along with other chemokines can contribute to the inflammatory cascade in Alzheimer's disease, potentially exacerbating neuronal damage and cognitive decline. That has been shown. What you're seeing here is a trajectory where you start getting amyloid deposits, leads to inflammation, which is very important, and MCP1 and IP10 are part of that. Then that leads to tau pathology, neuronal dysfunction, and then obviously cognitive decline that we visited. So these are controls. These are all age-matched controls. Our Alzheimer's individuals are at about 70.1 is the average age versus 69.8 or 69.2 in the controls. Sex-matched, age-matched. We have also looked at social determinants and so on, educational level. Everything is matched in this case. And you can see the levels of MCP1 are significantly up and also of IP10. So controls are 17 and 80. We are adding more numbers to this right now. This is looking at GFAP and neurofilament light. So higher GFAP and NFL levels are linked to increased amyloid plaque density, cognitive decline, and worse performance on neuropsychological tests in Alzheimer's disease patients. So that's known. So we are looking at GFAP and NFL. And then before I show this data, I just want to say that we are lucky that we were able to establish these diagnostic markers in the lab. You send these diagnostic markers out right now. A company called C2N charges $1,400 per person, and so and biomarker field was lagging. It's now picking up, and that's the main difference. That uh, by the time a person sees a neurologist, the horse has left the barn. So the analogy I was discussing with Dr. Moksha earlier, that if you have a certain level of fasting glucose, your A1C is up, you're not going to wait till you have kidney damage or any retinopathies. So you start treating, but that doesn't happen in neurology because there is no screening, there's no biomarkers available. And as I said, 80 biomarkers appear 8 to 10 years before the diagnosis is made. So there's a big debate that this type of thing should go into the hands of PCPs and not because, first of all, neurologists, I can tell you from my colleagues, are so overwhelmed with in the clinic. So this is the A beta 42 to 40 ratio. It is significantly down, and that's the hallmark. What that tells me, when you see a lessening of this ratio in the plasma, it's sticking more in the brain, and that's why these numbers drop. This is the tau phosphorylated on threonine 217 and threonine 181. These are absolutely hallmarks of phosphorate tau, and now there's an FDA approved, just came out a few months back for p-tau 217, and they are saying it is as good as a CSF measure or even a lumbar, even a PET scan. So this, we are very lucky that we were able to establish these assays in the lab here. And then there are some, I won't go through this data because this has to do with lecanemab and ARIA and so on. Then we have looked at these individuals, what happens at the level of the microbiome. We are detailing it out. As you can clearly see, this is the control. This is the AD cluster, and there's the control. Same age, same gender, everything same, but clearly the microbiome is very distinct in these individuals. As I was talking about, because we wanted to know and we had that previous knowledge from the gut microbiome components in the butyrate producing bacteria, we initially looked at the phylum, and you're looking a loss of Bacillota. So Bacillota was previously referred to as firmicutes, if anybody knows this, and it has the largest compendium of butyrate producing communities, and that is changing, so that's the Bacillota. Then you have Bacterodota, Actinomycota. So that's the phylum distribution that is changing. We looked at firmicutes, or the Bacillota, and that is significantly down in these individuals compared to the controls. The next part is FB ratio. So FB ratio is also telling or comments on the diversity of the microbial composition, and that is again down in the AD patients. So it clearly started to show that there are differences, and we are detailing these differences out. One of the most recent data is to show we could see the differences in butyrate producing families, and this is the Clostridiaceae. What T means it's trending. It did not reach significance, but as we add more numbers of patients, we'll probably zone in on that. Lactinospiraceae and Oscillospiraceae. And we are also doing what is called as whole genome shotgun sequencing. So we are detailing it out at the level of strain and species all the way. And the end game, hopefully, is to be able to come up with evidence-based probiotics. We are also going to use a tributrin type of approach, and we are right now actively discussing what's going on. So this is the most recent data, not published yet. There's more things happening. We are also doing a metabolomic scan on that. We track about 1,700 molecules. We are looking at persistent organic pollutants that are present in these individuals. And as you know, importantly, there's a big news that trichloroethylene, which is a dry cleaning molecule. There's a map of trichloroethylene of pollution in the US. They could superimpose the Parkinson's map completely on it. And so we are looking at it now in many different ways. So in conclusion, the gut microbial dysbiosis marked by a loss of beneficial butyrate producing bacteria plays a major pathogenic role in the development of Alzheimer's disease, and that the gut microbiome is a potential significant modifiable factor that can be targeted for the development of preventative and therapeutic strategies. I think prevention is really, in this case, way better than cure. So this is where we are. This is the team, and that's the funding. So thank you very much. Yes. It supports butyrate growth. Well, it does affect the microbiome. That's known. Right. Butter. It's
non-heat.
well, I that's a good point. Need to really analyze. Next time I'm going to send a sample out. We are getting this analyzed anyways. So, but clearly it's there in dairy, for sure, and in rich. I mean, it's enriched nicely. The flavor part is through tributylin, so it's there very much. But eating your greens with the right kind of bacteria is a key thing to do, because that's what's going to give you that. Yeah, and then plant-based, yeah, which is going to, because that's, we have, in fact, one of the earlier individuals who did that work is sitting right behind you. She did the butyrate synthesizing. So we identified there are specific bugs that have butyrate synthesizing pathways, and they absolutely can give you a pretty significant component of butyrate through your diet. So which is seems to be clearly important.
So recently there was another publication about shingles vaccine preventing dementia, or people who had more, I guess, exposure to actual shingles or zoster had a higher risk. It was more of a observational study. So so I was just curious about it. And also I know 1 in 4, if you have like 1 in 4 have the ApoB mutation.
Going forward, yeah,
Yeah.
homozygous.
How does that tie with
That's a great, yeah. So it's the well-known genetic risk factor for Alzheimer's. If you are ApoE4 homozygous, both alleles are ApoE4. But again, in there, about 25 to 30% people who are ApoE4 homozygous get Alzheimer's, but that means 65 to 70% don't. So there are two papers in Science. What they did was they examined this concept of microbiome. So there is an animal model which is ApoE4 knocked in and tau, and they get clockwork Alzheimer's. Their brain has huge pathology and everything. When they derived these animals on a germ-free background, no Alzheimer's. So that was one. Then they went back to the conventional animals, put them on a truckload of antibiotics, which we cannot do for people, and again markedly mitigated the evolution of Alzheimer's. And the bottom line is that even if there is a genetic disposition, that requires the right kind of microbial things to manifest. So this is very important. And that's why prevention, yeah, because as I said, ApoE4, although is an extremely well-known, strongest genetic risk factor, not every person gets it. So the hope for the people will get scared when they get. We do the screen before giving lecanemab, which is the immune therapy, which removes plaques, because there's a very significant side effect called ARIA, A-beta related imaging abnormalities. And ApoE4 homozygosity somehow predisposes them to develop those side effects much more. And so that's why they are screened. And if they are homozygous, they are basically advised that that's. But even if they still want to, they are given. They're counseled that you have this predisposition. So yes, that one in there is. But if you look at the overall population of Alzheimer's or Alzheimer's disease-related dementia, it is predominantly sporadic, which they don't have ApoE4. In fact, we are just finishing a large retrospective analysis of the Cosmos database, which is de-identified EPIC data using all the ICD-10 codes and everything. And what we are finding is that it is remarkable how things like AUD, alcohol use disorder, depression, fragmented sleep issues, or they become like a nexus that is connected with. We are also trying to, I did mention this, and we are finding differences. We will see early onset, which is now on the rise, and late onset. So late onset is classically 65, 70 and above. These individuals are between 55 and 65. They are developing this. And that's why we are doing persistent organic pollutants. If there is something there in the environment that may be driving this process. The microbiome looks very different in early versus late. Forget about control. But we need more people. That's the study that's ongoing. So these are bringing different challenges. But
so there is the decrease in the butyrate.
Right.
It's common for many of these metabolic disorders.
Absolutely. Like type 2, that's what I showed that.
So are they individual? How are they different? What way are they different in these different disorders?
That's a great question. So there are nuanced differences. So if you look, these are not the same species or strains that are changed. Not only that, ultimately there are bacteria that use different substrates to make, like the predominant substrate acetyl CoA, which is utilized by a large component. But ultimately, that's why not just looking at the structural changes in the bacteria, but the functional outcome, like measuring butyrate levels, becomes critical because you might get numbers dropping because there could be so that's something that is not yet well appreciated. Then there's another component to that. You are also, these butyrate producing bacteria are called as secondary feeders. There are primary feeders. So that's why you'll have probiotics like lactobacilli, which are not making, but they support butyrate producing bacteria because whatever they make products that the butyrate producers are using to proliferate and grow. So it's a community effort, literally. That's why in my mind, usually one type of bacteria is not going to make a big difference. If you have a probiotic that has 10, 12, 14 strains as a collective, that has a far better outcome. But coming back to your point, that is an enigma. But clearly, we see different levels of butyrate. There are high butyrate producers. There are in between there because they get in a way classified as butyrate producers. But then we are trying to now very strictly connect it to the functional output rather than just the structural changes in the microbiome. And I'm almost certain that we are going to start seeing differences at that level. That even within, they're all cricket players. Somebody's a really good batsman who is with you, and that's helping you. So that's going to be, but that's been sorted out. But that is a good point. Otherwise, loss of butyrate. That's why I showed type 2 also has it. Alzheimer's has it. But I'll bet you're dying that now the question that you're asking is, and maybe we'll talk more, is there, nobody has looked, I mean, we are trying to do through ICD-10 codes, is there a higher occurrence? So if you take all the G30, which is the ICD-10 code for Alzheimer's, how much is type 2 diabetes a component in that population? Are they the predominantly represented or alcohol, whichever way? So trying to understand that. And the other thing is for even lecanemab, people don't respond. Is it, I don't know what is the state of their diabetes? Is it because of that they're not responding, or is it the microbiome? So some of the very important issues.
So in these animal models that they have any change to the liver, or the insulin resistant, or the other components that develop?
Oh, great question again. We did not see any, we just looked at fasting glucose. We did not actually did that. But what is known is your liver plays a significant role in turning over A beta peripherally. And looks like when you are building plaques, your liver is stuttering. It's not clearing A beta. In fact, there are very important protease called neprilysin which is hepatic, and they could see that in Alzheimer's postmortem in the livers that was down, and the A beta was accumulating. Animals also accumulate A beta when they are developing the Alzheimer's. So not yet gotten to that point, but liver is clearly affected. That's why alcohol. So we could clearly see that alcohol fast-tracked on this 18-month model. So what we saw at 18 months is happening at about 10 or 12 months. So clearly alcohol was pushing it. And one of the things that alcohol, and that's the grant which was scored well, what we could see was we could see A beta staining going up strongly in alcohol-fed animals. So liver is clearly a component of this. And what you correctly said in the introduction, it is a gut-liver-brain axis. So we have to absolutely look at that. By the way, we have published that tributrin protects the liver beautifully against alcoholic liver disease. Again, by particularly affecting, we tracked key gene, carnitine palmitoyl transferase, which is required for beta oxidation, which was suppressed by alcohol, was kept open by butyrate, and it cleaned out the liver nicely. But insulin resistance is an important part that we have to know.
Question online.
Yeah.
So I have a quick
Lorraine,
question.
it's a question from Swapna.
Yeah,
Two
so
questions. For
I can
animals
take
of
that.
both sexes studied.
That's a great question. There is sexual dimorphism in animals with regards to Alzheimer's. There's also there's a higher numbers of women who get Alzheimer's compared to men. In the animal model, the 3XTG, I'm glad you pointed that out. They're all females because the males do not develop. It's very erratic the way it happens in males. So it is. I'm glad this question was raised that there is sexual dimorphism. Oh, that's Dr. Winters. Okay. So that's a very important point. We do see that even in people, women get there's almost twice the number of women compared to men.
They're non.
Yes, yeah. No, but even after balancing that out, so that what is called propensity score matching and everything is done, women clearly are more at risk. Thanks. And then the question is, okay, from individual therapy for individuals to assess how far away okay, that's a great question. We are actively doing this observational study. We are also starting a couple international collaborations, one with Chile and in South Korea, and that is going to be funded by the Davos group, trying to figure out. Once we realize, given because the microbiome issues are contextual, you're absolutely right. We have to at least, we should have an answer, hopefully in the next 2 to 3 years, in the population that we encounter here in this geographical location. But hopefully soon. I mean, the advantage of tributrin was we did an end run. We did not go after some species of butyrate producing bacteria and try to add back. Tributrin, we did an end run by saying we'll give the product that we are seeing to be down, and that worked. But it also protected, that was a serendipitous finding, that it protected the butyrate producing bacteria. So that's where we are. I hope I've answered your question, but we are kind of inching in on that, and hopefully at least 4 to 5 years, I guess.
So these
Got it. Thank you. That helped.
microbiome that you made, notoriously transient.
What?
Is this going to be like if you keep giving it, keep on?
We have NIH funded study that's ongoing right now. We have these are in the context of HIV. So we are baseline, and they're given for 6 months probiotics. It clearly changed many things. With the 12-month point is coming because the probiotics are stopped. We want to see if it has washed out or reverted back, which means whether you have to lifelong continue or whether you're able to truly, but my guess is we may have to keep the pressure of probiotics on for a long time because your bugs tend to like, go back to where they were to begin with. So, yeah, thanks.